Have you ever wondered what happens inside your body when you take a pill? It's fascinating. This journey of a drug within our body is called pharmacodynamics. In simple terms, pharmacodynamics is the study of how drugs affect our bodies. It's like a complex dance between the drug and your body's cells. This dance leads to changes that help you feel better. Drugs work by targeting specific parts of our cells. Think of it like a key fitting into a lock. The drug is the key, and the lock is a specific part of the cell called a receptor. When the drug binds to the receptor, it triggers a series of events that lead to a therapeutic effect. For example, a painkiller might bind to receptors in the brain that block pain signals. But it's not just receptors that drugs interact with. They can also affect enzymes, ion channels, and even tiny particles called protons. Each of these interactions plays a crucial role in how a drug works. Understanding these interactions helps doctors choose the right drug and the right dose for each patient. In the following sections, we'll explore each of these drug targets in more detail. We'll learn how drugs work at a molecular level to produce their effects. By the end, you'll have a better understanding of the amazing world of pharmacodynamics. Let's start with receptors. These fascinating structures play a crucial role in how our cells communicate and respond to various signals. Receptors are like tiny locks found on the surface of our cells. They are essential for transmitting information from the outside to the inside of the cell. They are very specific, meaning that only certain drugs can bind to them, just like only the right key can open a specific lock. This specificity ensures that the correct signals are sent and received. Drugs that activate receptors are called agonists. These drugs mimic the natural substances that normally bind to the receptor. They're like the right key that turns the lock and opens the door, triggering a response in the cell. This response can vary depending on the type of receptor and the cell it is located on. For example, albuterol is an agonist that activates receptors in the lungs, opening up the airways and making it easier to breathe for people with asthma. This is a life-saving mechanism for many individuals. On the other hand, antagonists are drugs that block receptors. They prevent the natural substances or other drugs from activating the receptor. Imagine putting the wrong key in a lock. It won't open the door and might even jam the lock. This is how antagonists work, by blocking the receptor and preventing any response. Antagonists work similarly, preventing other molecules from binding to the receptor and blocking their effects. This can be useful in conditions where blocking a receptor can prevent harmful effects. For example, antihistamines block histamine receptors, preventing the allergic reactions caused by histamine. This helps reduce symptoms like itching, swelling, and runny nose. The strength with which a drug binds to a receptor is called its affinity. High affinity means the drug binds very tightly to the receptor. A drug with high affinity is like a key that fits snugly into the lock and turns it easily. This ensures a strong and effective response. This means it's effective even at low doses. High-affinity drugs can be very potent, requiring only small amounts to achieve the desired effect. The relationship between the dose of a drug and its effect is called the dose-response relationship. This helps in determining the optimal dose for achieving the best therapeutic effect. It's like turning up the volume on a radio. The higher the dose, the stronger the effect, up to a certain point. Beyond this point, increasing the dose may not increase the effect and could cause side effects. Sometimes receptors can become less sensitive to a drug over time, a process called desensitization. This can happen with repeated use of the same drug. It's like using the same key repeatedly. Eventually, the lock might become worn out and harder to turn. This means the drug becomes less effective. This is why some drugs become less effective over time, and doctors may need to adjust the dose or switch to a different drug. This ensures that the patient continues to receive the best possible treatment. Next, let's move on to enzymes, which are truly fascinating components of our biological systems. Enzymes are like tiny machines within our cells that speed up chemical reactions, making processes that would take much longer happen in mere seconds. They're essential for a wide range of bodily functions, from digesting food to copying our DNA and even repairing damaged tissues. Some drugs work by interacting with these tiny machines, either enhancing or inhibiting their activity to achieve a desired therapeutic effect. Enzyme inhibition is one way drugs affect enzymes, essentially putting a brake on the enzyme's activity. Imagine throwing a wrench into a machine's gears. It stops working, or at least slows down significantly. Similarly, enzyme inhibitors bind to enzymes and block their activity. preventing them from catalyzing reactions. For instance, statins, drugs used to lower cholesterol, work by inhibiting an enzyme involved in cholesterol production, thereby reducing the amount of cholesterol in the bloodstream. Competitive inhibition is a specific type of enzyme inhibition where a drug competes with the enzyme's natural substrate for the same binding site, effectively blocking the substrate from binding. It's like two keys trying to fit into the same lock. Only one can bind at a time, making it a competitive process. The drug's effectiveness depends on its concentration and affinity for the enzyme compared to the natural substrate, determining how well it can compete. Conversely, some drugs activate enzymes instead of inhibiting them, enhancing their natural activity. It's like adding oil to a machine, making it run smoother and faster, improving its efficiency. Enzyme activators enhance the enzyme's activity, increasing the rate of the chemical reaction it catalyzes, which can be beneficial in various therapeutic contexts. Some enzymes have allosteric sites, which are like secondary binding sites distinct from the active site where the enzyme binds its substrate, offering additional regulatory control. Drugs that bind to allosteric sites can either enhance or inhibit enzyme activity, acting as allosteric activators or inhibitors, providing a nuanced way to modulate enzyme function. Prodrugs are a fascinating example of how drugs can utilize enzymes to become active only when needed. These drugs are inactive in their administered form but are converted into their active form by enzymes within the body, ensuring that the drug acts precisely where and when it is needed. It's like a delivery service. The prodrug is the package delivered to your doorstep, and the enzyme is like the person who opens the package, revealing the active drug inside, ready to do its job. Now let's talk about ion channels. These are like tiny gates embedded within the cell membrane, controlling the flow of charged particles called ions in and out of the cell. These ions are crucial for many physiological processes, including nerve impulse transmission and muscle contraction. Some drugs work by blocking ion channels. It's like closing a gate, preventing ions from passing through. Local anesthetics, for example, block sodium channels, preventing nerve cells from sending pain signals to the brain. Other drugs modulate ion channel activity without completely blocking them. They can make the channel open or close more easily or change the duration of its opening. It's like adjusting the tension on a gate spring, making it open or close with more or less force. Ion channels can be voltage gated, meaning they open and close in response to changes in the electrical potential across the cell membrane. Or they can be ligand gated, meaning they open when a specific molecule, like a neurotransmitter, binds to them. Similar to receptors, ion channels can also undergo desensitization. Continuous exposure to a drug can make the channel less responsive over time, requiring a higher dose to achieve the same effect. Finally, let's delve into the fascinating world of protons and their significant role in our bodies. Protons are tiny, positively charged particles that are crucial for maintaining the acidity or alkalinity of our body fluids, a balance that is vital for our health. This balance, known as pH, is essential for many biological processes, including enzyme function, nutrient absorption, and overall cellular health. Proton pump inhibitors, commonly known as PPIs, are a group of drugs that reduce the production of stomach acid, providing relief from various acid-related conditions. They work by blocking an enzyme called the proton pump, which is responsible for pumping protons into the stomach, making it more acidic. By inhibiting this enzyme, PPIs effectively reduce stomach acidity. PPIs are commonly used to treat heartburn, ulcers, and other acid-related disorders, providing significant relief and improving quality of life for many patients. pH regulation is crucial for drug absorption and distribution in the body, influencing how effectively medications work. Some drugs are absorbed better in acidic environments, while others are absorbed better in alkaline environments. This variability can affect the efficacy of the medication. Understanding the pH of different body compartments helps in predicting drug absorption and optimizing drug delivery, ensuring that medications reach their intended targets effectively. Proton gradients, the difference in proton concentration across a membrane, are essential for energy production in our cells, particularly in the mitochondria. Some drugs can disrupt these gradients, affecting cellular energy production and potentially leading to adverse effects. Proton channels are specialized proteins that facilitate the movement of protons across cell membranes, playing a critical role in maintaining cellular function. These channels play a role in various physiological processes, including acid secretion and pH regulation, which are vital for maintaining homeostasis. Maintaining acid-base balance is crucial for overall health, as it ensures that our body's systems function optimally. Imbalances in pH can lead to various health problems, including metabolic disorders, respiratory issues, and even cardiovascular diseases. Drugs can influence acid-base balance, and understanding these effects is essential for safe and effective drug therapy, ensuring that treatments do not inadvertently cause harm. Understanding how drugs work at a molecular level is crucial for developing effective treatments and improving patient outcomes. It allows doctors to select the right drug for the right patient, determine the appropriate dose, and minimize the risk of side effects. By understanding the concepts of pharmacodynamics, we can appreciate the complexity of drug action and the importance of personalized medicine. Just as each lock requires a specific key, each patient may respond differently to a drug based on their individual genetic makeup, lifestyle, and other factors. Pharmacodynamics helps us understand why some individuals experience side effects from certain drugs while others do not. It also sheds light on why some drugs are more effective in certain populations than others. As we continue to unravel the intricacies of drug action, we pave the way for developing safer, more effective, and more personalized therapies. This knowledge empowers both healthcare providers and patients to make informed decisions about their health. Remember, this is just a glimpse into the fascinating world of pharmacodynamics. By understanding how drugs interact with our bodies, we can unlock the full potential of medicine and improve human health.